Hello everyone, welcome back to Affordable Tutor, where we make education simple and exciting for everyone. So in today's video we are going to discuss about Swiss models, which is a web-based service that automates the process of homology modeling in protein structure predictions. It means that it uses the known 3D structures of similar proteins, which we call it a template, to predict the structure of target protein. So it is developed by a computational structural biology group at the SIB, Swiss Institute of Bioinformatics, and the biocentrums of the universities of Basel. So it can be used by many field researchers like molecular biology and biochemistry and drug discovery, so forth. As you can see in the window that is currently opened, it said that homology modeling servers. So what is homology modeling? It's like working on the principles like proteins that have a similar sequence tend to have a similar 3D, like three-dimensional structures. So they work on the principle of that. So it also has its repository, and as per they said, they models each and every sequence in the UniProt. They are modeling it every week. So if you just want to look for it, just type in and it will just give you your protein structures that has been predicted by Swiss model. So I'm just writing EGFR, that's a protein, a transmembrane protein, and here it just give me that protein predictions that they already predicted. So here you can see homology model, experimental structures, and so forth. So there is concept of experimental structures and predicted structure. Experimental structure is basically when you do X-ray crystallography and NMR, nuclear magnetic resonance, for proteins, 3D model structure, and one is the predicted. So here I'm just clicking on the start modeling. So here we have the input field for our protein sequence, and here you can see it support FOSTA file, and we have like paste your sequence, or you can upload it here as a FOSTA file, or you can just use the UniProt ID here. But we have here like examples for single target example, multiple target examples. You can just like it just give you the overview or a guide for uploading a sequence. And here we have another one like target template alignment. If you have a target and template and you're doing the alignment for each of them, comparing them. So here you can see we have like two target and the template, and you're doing the alignments. As you can see, there's still we have a difference in the amino acid, and you just click on start building a model. So it will create and predict a model for you. As I have told you that if you have the template, you just have to upload it here for the like when just put your sequence here. Like for example, I'm putting it here sequence, and you can just upload your template and give it a title, email, so and start building your protein. Another one is the D project view, which you have to just upload a deep view project files, as you can see here in the example as well. You just can to download it and upload it here. So for we'll be not using this. We will be using the sequence one, as you can just put your sequence here, project title of your project, and like I'm just using the single click on it. Here you can see the project title. The email is just for like reminder, like when the job is complete, it will just give you the email a reminder that your job has been done. Here is one thing that you must need to understand, that you can look for like search for template, or you if you already have it. So if you don't have it, just go and search it. But if you just leave it to the Swiss models, it will use the auto modeling feature for you, and we'll just have to click the search model as well. So let's just click on it, and as you can see, it's looking for the template for your proteins or for your sequence. I have already here you can see it has start working, and I've already execute this for you, so we can just quickly go for that. Here you can see here's the results of when it's completed. It has 450 templates so far, and as you can see we have identity 99.34, and it's X-rays, mean it's experimental structure with a 2.0 angstrom, and the rest is here like quaternary structures, and here you can see the rest of things and sequence similarities, alignment. And if you want to look into the deep, like a deep view, you can just download the zip file so far, and here it is what it is. So I'm just putting the target again. Also we just explore the Bull's model, which is like it will just use the auto modeling feature of Swiss model, and yeah, it will be running like that. Your models will appear here when ready. So I already ready it for you. Like you can see, they have found 50 template and model 2, like the one that are best. And here you can see 2ED, it's a similar sequence proteins compared to this target protein. And here you can see everything about your protein. You can just film it for you, like, and it will give you the confidence of each and every amino acids and so forth. And that's the another one, and that's for today, that we have now predicted a protein structures for from a sequence of a protein. So if you have it, just go for it and do it. Sometime like we predict a structure of a mutant proteins, like we induce a mutation in it and then predict the structures, and to see superimpose both the proteins on each other and just look for the differences. And for the structure assessment, we'll be doing it in the deep in our next video, which is here the options. Will be exploring the different sci-fi angle Ramachandra review. And here you can see when the job is done, it just appear here like the sessions in the sessions. So you can just delete it, or if you visit it again, it will be just here, just to click on it. Like if I click on that example, as you can see, it has been like a summary and a template and the model that is already been modeled so far for you. And here you can see when the job is done, it just appear here like the sessions in the sessions. So you